Sir, start, sir.
Okay. Good morning, students. Welcome to Botany webinar classes conducted by Telangana Residential Educational Institution Society. This is Srikanth, lecturer in Botany from Sangam Lakshmbai, TSRJC, Vikarabad. Students, in previous sessions, we discussed about genetics topic. Okay, genetics unit. In this unit, we discussed about some various different types of topics. Those are introduction of genetics and Mendel inheritance laws, and as well as we discussed about monohybrid cross, and law of dominance, and Gregor John Mendel, father of genetics, a Mendelian concept, and some terminologies of genetics, and advantages of pea plant, mono and dihybrid cross, test and brag cross, incomplete and codominance. These are the contents we discussed. These are the contents we discussed in previous sessions. Okay, in previous sessions we discussed these are the contents. Okay, students, this is the last session for genetics topic, for genetics unit. In this session, we are going to discuss about chromosomal theory of inheritance and mutation. Chromosomal theory of inheritance and mutation. These are the two topics we will discuss today. Okay, okay, students. First, what is chromosomal theory of inheritance? See, so we know that Gregor John Mendel, he also popularly known as father of genetics, because he experimented on pea plant and he did some hybridization experiments and proposed some inheritance laws. But Mendel died unrecognized man. Later, in the year 1900, three scientists rediscovered Mendel's results. Three scientists. Those were Hugo de Vries, Corens, and Van Schemark. Okay, already given about these three scientists in introduction class. Those were de Vries, Hugo de Vries, and Corens and Schemark. These three scientists were rediscovered the Mendel results, because at the time advancement of microscopy, means advanced microscopes were discovered at the time of de Vries, Corens, and Schemark. Also, scientists are able to carefully observe cell division due to the advancement of microscope. We know that electron microscope discovered by Nall and Ruska, they both were discovered the electron microscope. Nall and Ruska. They both were discovered the electron microscope. After the discovery of the electron microscope, most of the cell properties observed by scientists. In that, one of the cell property is the cell division. Cell division observed by first time Rudolf Virchow. We know that. We already discussed in first year. Rudolf Virchow observed the cell division. According to him, omnicellula acellulae means the new cells were generated from pre-existing cell. The new cells were generated from pre-existing cell according to Rudolf Virchow. This observation due to the electron microscope only. And this led to this discovery of structure in the nucleus. And this led to the cell division discoveries lead to the discovery of structures in the nucleus. Those structures are appeared to double and just before each cell division. What is this? The structures which are present in the nucleus, double. When they are in double, just before each cell division. These were called as chromosomes. Here, chromo means color. Zomes means bodies. Chromo means what, Nana? Color. And zomes means bodies. Which means the meaning of chromosome is color body. The chromosome's meaning is color bodies. The chromosomes were discovered by first time Walter Fleming. Who discovered the chromosomes first time? Walter Fleming. And later, Walter Sutton and Theodore Boveri. They both were noted that the behavior of chromosomes was parallel to the behavior of genes. Excellent point this is. Observed by Walter Sutton and Theodore Boveri. According to Walter Sutton and Theodore Boveri, what is that? The behavior of chromosome was parallel to the behavior of genes, which means the chromosomes and genes both are performing same properties. What is this? The chromosome and genes both are performing both properties, performing in same nature, according to Walter Sutton and Theodore Boveri. Later, they both were proposed chromosomal theory of inheritance. Which means, according to chromosomal theory of inheritance, the genes are located on the chromosome. The genes location part is called as loci. What is that? Loci or locus. Locus or loci. The genes present on the chromosomes. If genes are responsible for the inheritance, the chromosomes also responsible for the inheritance. If genes are segregating, the chromosomes also segregating. If genes are dividing, the chromosomes also dividing. which means chromosomes was parallel to the behavior of genes. The chromosomes are equal to the genes' behavior. That is why chromosomal theory of inheritance. So most of the people think that thought that the genes are responsible for the inheritance. The genes was responsible for inheritance, but they did not identify the gene's location. Walter Sutton and Theodore Bowry located the gene's place on the chromosomes. That place is known as locus or loci. Later, they both were proposed that chromosomal theory of inheritance, which means the chromosomes also responsible for the inheritance along with the genes. Okay, so see, students, here two columns are here. The column A, column B. In column A occurs in pairs. Column B occurs in pairs. Column A segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. What is that? Segregate at the time of gamete formation. These pairs are separated during the formation of gametes. These pairs are separated during the formation of gametes. That only one each pair is transmitted to a gamete. Only one gene is each pair is transmitted into a gamete. Come to here. Segregate at the time of gamete formation, and only one of each pair is transmitted to a gamete. Both are similar. So this is also similar. This point also similar. Third point, independent pairs segregate independently each other. Independent pairs. If the pairs are in independent manner, they are separated, means segregated. They are showing their characters independently each other. They are showing their characters independently each other. And here, one pair segregates independently one of other pair. This point also same. Both are in similar pairs, similar points. Students, according to this column, can you find out which is gene and which is chromosome? Which is gene column, which is chromosome column? Can you find out? Yes, we can't, because these all characters are shown by genes and chromosomes. That is why we can't separate the genes and chromosomes by these characters. So this is known as chromosomal theory of inheritance. Chromosomal theory of inheritance. Students, who proposed the chromosomal theory of inheritance? Walter Sutton and Theodore Bowry. Who are they? Walter Sutton and Theodore Bowry. Okay, so they were taken the information from. They were taken the information from three scientists which were rediscovered the Mendel's results. Those were Hugo de Vries, Orens, and von Schemark. From them, Walter Sutton and Theodore Bowry taken the information and proposed the chromosomal theory of inheritance. According to them, chromosome and genes both are similar in nature, both are behaving same in manner. Okay, students, here two columns are here, students. Chromosomes. In possibility one long orange and short green chromosome and long yellow and short red chromosome at the same pole. Okay, here one long orange and short green, and this side long yellow and short chromosome at the same pole. When it is during the meiosis anaphase time. This is a possibility too. One long orange and short red chromosome and long yellow and short green chromosome at the same pole. It is also meiosis 1 anaphase. Okay, students, according to this picture and this diagram, here one column is located at the left hand side, one column is located at right side. Okay, left side and right side. See, during metaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate independently each other. What is that? The two chromosome pairs can align at the metaphase plate independently each other. Okay, so and to understand this, students, compare the chromosomes of 4 different colors. Here 2 colors, here 2 colors. 4 different colors of chromosomes are here: orange, green, yellow, red. Orange, green, yellow, red. 4 different colors of chromosomes are here. So compare the chromosomes of 4 different colors in the left and right columns. Left and right columns. In the left column, and take possibility 1. In the left column, orange and green are segregating together. Orange and green are segregating together. But in the right hand column, the orange chromosome is segregating with the red chromosome. The orange chromosome is segregating with the red chromosome. Sutton and Boveri argued that the pairing and separation of pair of chromosome would lead to the segregation of a pair of factors they carried. Okay. So this phenomenon argued by Sutton and Boveri, that the pairing and separation of a pair of chromosomes would be lead to the segregation of a pair of factors. Pair of factors they carried. Okay, they carried. So here the chromosomes carrying the factors, nothing but genes. Based on these genes, they are segregating and separating. So Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Later, Sutton and Boveri, they both were compare these phenomenon with the Mendelian principles. Later, proposed the chromosomal theory of inheritance. Okay, so who proposed the chromosomal theory of inheritance? Sutton and Boveri. They both were proposed the chromosomal theory of inheritance. Okay, students. Next, so linkage and recombination. By the chromosomal theory of inheritance, later Thomas Hunt Morgan, shortly known as T.H. Morgan. Thomas Hunt Morgan experimented on one fruit fly. That fruit fly name is Drosophila melanogaster. Drosophila melanogaster is a fruit fly. And later, he noticed that linkage by the experiments on Drosophila melanogaster. According to Thomas Hunt Morgan, the linkage means the physical association of two genes. The physically association of two genes on homologous chromosomes. The physical association of two genes which are present side by side. This linkage leads to recombination. The linkage leads to recombination. So the linkage of two genes, the mixing of two genes together, the recombination takes place. Due to this, non-parental combinations are developed in the next generation. Non-parental combinations are developed in the next generation. That is known as recombination. The recombination leads to linkage. Okay, so recombination leads to linkage. So the linkage leads to recombination. That means linkage means the physical association of two genes. The physical association of two genes. Due to this, non-parental combinations are developed in the next generation. Due to this, non-parental combination developed in the next generation. That is recombination. So by the recombination, evaluation takes place in the world, in the organisms. In organisms, in traits, evolutions are takes place. Means diversities are takes place in the organisms due to the recombination. If there is no linkage, there is no recombination. If there is no recombination, there is no diversity. There is no diversity, which means the diversity takes place by the linkage of two genes on the chromosomes, especially in animals. Okay, so the due to the linkage process, due to the physical association of the two genes, new individual will be developed in the next generation. For example, capital Y, small y combined together form a capital Y, small y plant in next generation. Okay, so imagine this is a capital Y F1 generation. In F1 generation, the capital Y, small y plant will be developed. Okay, students, this is the plant showing some variations to the parents. The parents are pure in nature, but the F1 generation is the heterozygous in manner. The F1 plant is showing non-parental character. The F1 plant is the recombinant plant. This recombination takes place due to the association of the two genes from the gametes. Okay, students, this is about linkage and recombination. Sometimes some changes are takes place in the genes. These were first noticed by Hugo de Vries. These were first noticed by Hugo de Vries. Okay, so in one plant, the plant is known as Enothera lamarckiana. Known as Lamarck primrose, also known as Lamarck's primrose plant. Enothera lamarckiana, also known as Lamarck primrose. The plant name is Enothera lamarckiana. The scientist was Hugo de Vries. He first observed the changes in the genes in one plant. The plant is known as Enothera lamarckiana, also known as Lamarck primrose. That change is known as mutations. mutations. What is mutation? The sudden change in DNA. Okay. The sudden change in DNA known as mutations. Sometimes mutations that takes place in offsprings in next traits. Due to this, some variations are present in the offsprings. Okay, that is mutation. The mutations were first time observed by Hugo de Vries. That is why Hugo de Vries is also known as father of mutations. Father of mutations, he was Hugo de Vries, observed in Inothera lamarckiana plant. The sudden change in DNA known as mutations. Sometimes mutations that takes place in DNA sequence. In DNA sequence, the sometimes changes that takes place. The sudden changes that takes place in DNA sequences in single base pairs. Okay, base pairs. Base pairs means what, Nana? Base pairs means what? The base pairs means simple. The DNA has base pair means the DNA has 4 bases: adenine, guanine, thymine, cytosine. Adenine is bond with thymine. Guanine is bond with cytosine. This one bond is called as one base pair. The adenine is the one base, thymine is the one base. Both are bonding together. That bond is known as one base pair. This is a one base pair. Sometimes in these base pairs, the mutations means changes are takes place. Changes are takes place. That changes is known as point mutation. Point mutation. Mutations arises due to change in a single base pair of DNA known as point mutation. For example, sickle cell anemia. Sickle cell anemia is a disease which is causes to RBC, red blood cells. We know that red blood cells are biconcave in shape. Biconcave in shape. If biconcave shape of red blood cells showing mutations, if biconcave shape of red blood cells showing mutations, they are turns into sickle shape. Sickle means kodavali. Kodavali, you know kodavali? Okay, so that is the instrument using for cutting the grass or some other plants. Okay, so that is sickle. The biconcave shape of RBC turns into sickle shape. That condition is known as sickle cell anemia. That condition is known as sickle cell anemia. Okay, students. So this is about your genetics topic. So, in this class, in today's class, the important points were point mutation, Hugo de Vries, Inothera lamarckiana. So who noticed the mutations first time? Hugo de Vries. In which plant? Inothera lamarckiana. Okay. So next one, the linkage. Who observed the linkage first time? Thomas Hunt Morgan. In which organism? Drosophila melanogaster, fruit fly. What is the combination? So this is a diagrammatic manner for the clear understanding about chromosomal theory of inheritance. This is the comparison of gene and chromosome. Both are similar in nature. And this one, Walter Sutton and Theodore Boveri, they both were proposed the chromosomal theory of inheritance. And this is the information in today's class. Okay, students. and take assignment, all of you. So please do your assignment, forward to your concerned lecturer compulsory. Okay, so please forward to your concerned lecturer. Take this assignment, all of you students, and very short questions and short questions. Most important short questions. These are for IPE purpose. Most important questions. Most important short questions. These are for IPE purpose. Okay, so short questions. First, who proposed chromosomal theory of inheritance in today's class? Sutton and Boveri. Define true breeding. Mention its significance. Second question. Third question. Explain the terms phenotype and genotype. What is point mutation? Give an example. What is the genotype of wrinkled phenotype of pea seeds? Short, very short questions these are. VSQs. Most important 2 marks purpose. Okay? Repeatedly asking questions in IP. These questions. Short questions. Define and design a test class. Explain the codominance with example. Explain the incomplete dominance with example. Explain law of dominance using a monohybrid class. Explain law of dominance using a monohybrid class. Mention the advantages of selecting pea plant for experiment by Mendel. Okay, so these are the most important questions and repeatedly asking questions in IPE. Okay, so okay students, and thank you very much. And I would like to tell the thanks to our secretary, Honorable Secretary Sir, Ramana Kumar Sir Garu, and Deputy Secretary Sir, Prasad Sir Garu, and all society members, and subject coordinator, Sarala Madam Garu. So, and given this wonderful opportunity, thank you very much. Thank you students for your cooperation, and all the best for your future. And I wish you all for your wonderful future, and good luck to all. Okay, thank you very much. So today's class over and genetics topic completed.
Srikanth sir, meeting close చేయండి, మీరు co-host ఉన్నారు.
మేడం
మీటింగ్ క్లోజ్ చేయండి, మీరు co-host ఉన్నారు కదా, క్లోజ్ చేయొచ్చు.
ఓకే మేడం. లేదు మేడం, ఒక్క నిమిషం నేను చెప్తాను. మేడం నాకు ఎండ్ కావటానికి రావట్లేదు మేడం, ఓన్లీ లీవింగ్ అని వస్తుంది. లీవ్ చేయనా?